Let's just give a minute. Let's get started. 国亮你全屏一下好吧？
我现在已经在全屏了，能看见吗？
OK 可以。你第一张吧。
第一张，这就是第一张。
Okay, so let's get started. Welcome everyone to today's Immune seminar. It's really my great pleasure to welcome today's speaker, also my friends, Dr. Guoliang Cui from German Cancer Research Center, DKFZ in Heidelberg. So a very brief introduction about Guoliang. So as I said, currently Guoliang working as a group leader, also a head of division in German Cancer Research Center in Heidelberg, Germany. So Guoliang actually complete his PhD training in biochemistry and molecular biology in Shanghai Institute for Biological Sciences, Chinese Academy of Science. And then following which Guoliang actually have a brief training and working experience in Department of Neuroinflammation Division in GSK in China from 2010 to 2012. After that, Guoliang actually continued to pursue his scientific career, training as a postdoc fellow with Susan Kirk in Yale in Department of Immunobiology. And then after from 2016, after he completed his postdoc training, he actually moved from US to Germany, working as a junior group leader in DKFZ, which is also German's Cancer Research Center in Heidelberg, Germany. And just by last year, he was promoted as a head of division, which is a permanent position in DKFZ in Germany. So Guoliang actually's group is particularly interested in metabolism in cancer biology related to the immune response. As we know that immune checkpoint blockade has achieved a huge clinical success in several types of cancer, but only a very few subset of population of cancer patient actually benefit from this, which actually Guoliang realized this suggests that we may be still at a very early stage in understanding the immunosuppressive nature of microenvironment in tumors. Therefore, one of the most important questions in the tumor biology field is to identify the factors causing T-cell exhaustion in the tumor. Based upon the previous work, Guoliang's group, which mainly focused on T-cell metabolism, use mouse melanoma models to identify the metabolic checkpoints regulating anti-tumor T-cell bioenergenic survival and effector functions. The long-term research goal of Guoliang's group in T-cell metabolism is to translate their findings made using the mouse model into clinical benefits. In particular, they are interested in addressing a few questions, including which lipid metabolites regulate human anti-T-cell effect function and survival, and what is the mechanism underlying lipid-induced T-cell immune suppression, and then finally, how to enhance human anti-tumor T-cell function by regulating the lipid metabolomic pathways. These future studies probably can lead to novel immune therapies by targeting T-cell intrinsic lipid metabolism. So I think a lot of us probably already know or have read Guoliang's scientific paper related to the T-cell metabolism, particularly in the lipid metabolism. These really groundbreaking findings actually lead to numerous awards during Guoliang's career, which actually including, but not limited to House Young Investigator Awards in Germany, George's Collins Prize, also Germany last year, EMBO Young Investigator Awards in 2020, and Lloyd Old Start Awards in 2021. And this year, he also got ERC Consolidator Awards in Europe, in Brussels. So I think without further ado, Guoliang, we are very much looking forward to your talk. Please take away.
Thank you, Frank. Thank you so much for your very nice introduction. Very happy to be here and to say older friends and also to make new friends here. So today, I would like to tell you about our research into T-cell exhaustion. I have two pictures here of cats. The right one lying flat, seems very tired and sleeping, and the left one is fighting. But no worry, I'm not going to switch my topic from T-cells to cats, but rather I find the diversity in the cat's behavior perfectly resembles the heterogeneity of the exhaust T cells. So for a long time, exhaust T cells have been considered to be the T cells that have been tired of chronic antigen stimulation and completely lost their effector function. But gradually, this opinion has been changed over the decades by the discovery of the heterogeneity of T cells. There are some cells indeed losing their effector function and become terminally differentiated, like this one, lying flat and give up. But there are also a few other subsets within the exhausted T cell population. So these subsets still retain a certain amount of T cell stimulus, and they still have effector function. So I hope that at the end of my talk, I could convince you of the similarity between T cells and cats. So when we talk about the T cell exhaustion, what exactly are we talking about? To answer this question, I think we needed to revisit the original definition of T cell exhaustion. So this is the earliest study that I was able to find of T cell exhaustion. It is from Professor Wolf, Signadro, the Nobel Prize laureates in 1996. He has a lot of landmark work in T cell responses to viral infection, and he mainly uses a virus called lymphocytic chorial meningitis virus, or LCMV. So there are quite a few strains of LCMV, but in general, these strains could be categorized into two classes. One class causes acute viral infection. The other one causes chronic infection. During acute infection, for example, with LCMV Armstrong, the antigen-specific T cells very rapidly expand from the effector T cells pool. And this virus could be cleared roughly within 1 week by the immunocompetent mice. And then 1 week later, after the viral clearance, the effector T cells pool starts to contract, and eventually leaving a small population of cells mature into memory phase. And this memory T cells persist a very long time, so they could maintain themselves through the homeostatic cytokine dependent self-renewal process. They're also able to differentiate into effector cells upon re-challenge. In contrast, during chronic infection, the T cells differentiate into a unique status called exhausted T cells. These T cells express high levels of inhibitory receptors and also reduce the effector molecules, which is very different from the effector T cells, memory T cells, or the naive T cells. But back to 1993, the definition of T cell exhaustion is a bit different from what we understand today. So in this landmark paper, it was interpreted that the antigen, the viral antigen persist as without the antigen-specific T cells were used up, and they almost disappear within 2 weeks. So this is the original definition. But anyway, regardless of this difference, so in the past 3 decades, the concept of T cell exhaustion has been evolving a lot. The T cell exhaustion does not only occur in viral infection, but also in tumor. I think to this audience, it's probably already common sense that the immunotherapies, especially checkpoint blockade-based therapies, have achieved huge clinical success. Therefore, it created a lot of enthusiasm, not only in academia, but also in the industry and the clinic. This immunotherapy is indeed effective in several types of cancers, but not in all of them. There are several possible explanations. First, many types of tumors do not have enough infiltrates because the therapeutic efficacy of the checkpoint blockade depends on the pre-existing CD T cells in tumor. If there are not enough responders, of course, we do not expect high therapeutic efficacy. Second, even T cells infiltrating into tumor, they differentiate into this unique status called exhaust T cells in both cancer and chronic infections. We certainly know cancers and chronic infections have different etiology, but they do share certain common principles. For example, the inflammation and also the antigen persistence. Accumulating evidence suggests that antigen persistence is a major driver of T cell exhaustion. These exhaust T cells turn on the expression of the inhibitor receptors, like the star molecule PD-1 here, and also a few others. And the decreased effector cytokine production, which is also accompanied by the altered metabolism. They also have problems in mitochondria. So all of this led to the poor control of viral infection of the tumors. Because T cell exhaustion is so important, many efforts have been devoted to understanding T cell exhaustion. So what do we know about T cell exhaustion by today? The first lesson that I want to share is exhausted T cells are not necessarily beta T cells. So in this paper from Professor Rafi Ahmed lab, so they found that although using antibody to block PD-1, PDR1 could revive exhausted T cells, but genetic deficiency of this pathway actually causes the mouse death. This deficient mice succumb to immunopathology. So therefore, the T cell exhaustion is kind of adaptation to chronic antigen in order to avoid immunopathology. Although these exhausted T cells are not so functional as their effector T cells peers, although they're not enough to achieve a cure, but still they're able to fight against either virally infected cells or tumor cells for a long period of time and eventually do something. The second lesson is these exhausted T cells, they're very heterogeneous. For example, they contain a population of T cells that are marked by the high levels of T cell 1, T cell factor 1, which is the marker of T cell stimulus, basically. They also turn on the chemical receptor CXCR5, and they reduce the expression of TIM3. So in this paper, again, from Rafi and also from a few other colleagues in this field, and I apologize if your work is not cited here. They found that the T cells, CD T cells, expressing high levels of CXCR5, which is actually dependent on the transcription factor T cell 1. So these cells are major responders to PD-L1 blockade. In contrast, the T cells expressing lower levels of CXCR5, they're basically responding poorly to PD-L1 blockade, which is also reflected by these statistics. Along the same thread, the exhausted T cells could be divided into different subsets based on these markers. In this paper from Henning's group, they used this marker SLEM6 and also another one, TIM3, to differentiate tell exhausted T cells into different subsets. And Henning's group found the T cells expressing high levels of SLEM6. This one, they have the capability to differentiate into different subsets. For example, these cells do not only self-renew themselves, but also give rise to T cells of different phenotype, suggesting these cells are precursor cells, and they have the capacity to repopulate the exhausted T cell pool. In contrast, the TIM3 high cells, which also express low levels of SLEM6, these cells are terminally differentiated, and they have poor capability to give rise to other subsets. These cells are also responding poorly to PD-L1 blockade. And furthermore, the discovery of TOX as an important marker of exhausted T cells provide new insights into this field. TOX expression marks a subset of T cells that are really terminally differentiated. These T cells also express high levels of inhibitory receptors like PD-1 and a few others. Overexpression of TOX also reinforce the exhausted T cell phenotype. But TOX is also required for the survival of these exhausted T cells, which I will mention a few slides later. Another point that I want to make is between the T cell 1 high stem-like T cells and the terminally differentiated T cells, which is marked by the TOX expression, there are also intermediate status. For example, the chemical receptor 63CR1 marks this intermediate T cell subsets, and they are actually doing a lot of important work in fighting against virus-infected cells and also the tumor cells. I think Wig will also give a presentation a few weeks earlier talking about this great work. So having said so much, we have this field has made a lot of progress in understanding T cell exhaustion. What else we do not understand? There are a few, for example, even until today, we do not fully understand the pathways leading to T cell exhaustion. And we need this information to figure out ways to reverse exhausted T cells. According to the work from various lab and Hanning's lab, exhausted T cells are epigenetically locked. So it's probably difficult to completely reverse these exhausted T cells, but to what extent we can reverse them? What is the balance point? And further, what is the relationship between exhausted T cells and also other subsets? All of these questions are very big questions. Even one of them is too big to tackle for a small lab like us. And also back in 2016, when I started my lab in Germany, we didn't know so much about the exhausted T cells. In all of this, most of this work that I introduced in the last 10 minutes actually didn't occur before 2016. So therefore, we decided to take baby steps, and we tried to answer a small question. We wanted to identify a marker of exhausted T cells and try to understand the pathways leading to exhausted T cells. That was our goal 6 years ago. To achieve this goal, we started from a very simple approach. We implanted B6 mice with the melanoma B16 cell lines, and this cell line is a bit different. It expresses the glycoprotein 3341. So this is epitope, which is naturally found in the virus LSM-way. And we got this cell line from Professor Hans-Peter Perscher in Freiburg. And then we implanted this tumor-bearing mice with T cell receptor transgenic P14 CD8 T cells. So these T cells could specifically recognize this epitope on tumor cells. So therefore, we are able to monitor the tumor antigen specific T cell response in this model. So this kind of comparable to OVA versus OT1 system. So there are a few systems like this. And then we take down the mice, we sort out this tumor infiltrating T cells from these tumors. This shows our getting strategy, and we send these T cells in tumor or in spleen for bunk RNA sequencing. The results were pretty clear, actually. We were able to identify more than 3,000 genes that were significantly increased in tumor infiltrating lymphocytes compared to their splenic peers. And more than, actually close to 5K genes were significantly decreased in TILs. We ranked them from number 1 to down the list, and we found a gene called RGX16 show up at the number 1 gene, which is turned on in the tails compared to splenic T cells. RGX16 actually shows comparable patterns with a few other genes associated with T cell exhaustion, like the famous molecule TOX, and also PDCD1, which encodes the star molecule PD1, and also STR4, a few others. RGX16 also shows the opposite expression pattern with the genes associated with T cell stimulus, like a cell which is the gene encoding the homing receptor CD62L and a few others. Here I wanted to highlight the TCF7, which encodes the protein TCF1. We also took one step further and looked at the expression of RX-10 in human tumor infiltrating T cells. We obtained the tissue sections from Heidelberg University tissue bank, and we were able to identify a few CD8 T cells in the colon tumors. Some of these CD8 T cells actually they express RX-10, not all of them, and we noticed that the RX-10 mainly localized in the cell-cell contact sites, suggesting RX-10 might be involved in cell-cell communication. We also noticed a lot of non-CD T cells express RX-10, suggesting that RX-10 might play a role beyond regulating T cell exhaustion. And after this sequencing analysis, we also look at the literature, and very interesting, we found two things, two very nice papers. One is from Professor Anana Goldiras. Her lab profiled the genes that are specifically turned on in tumor. This is cropped, but I can tell you this tumor in the spleen, and this is vaccinia viral infection and listeria infection. This is clone 13, and this is Armstrong. But in general, you can you just need to know that this is tumor, this spleen, and this chronic infection, this acute ones. And she found RX-10 is turned on in the tumor compared to spleen. And also a similar pattern was found in the chronic infection, which is very similar to the tox molecule. Another study from Dimerzan in Munich, Professor Dimerzan's lab found the tox, of course, is very important molecule in reinforcing exhausted T cell phenotype. And RX-10 is also not far away from tox. It's also one of the genes mimicking the same pattern of tox. So we were super excited when we thought this. So we decided to move on to look at the potential clinical relevance of this molecule. We dig out the previously published single cell sequencing data. So this is one from Professor Neil Hakon's group. And they've used the patient samples, the samples from melanoma patients who responded or not responded to PD-1 blockade and the CTLA-4 blockade. And we used data sets from the samples before the PD-1 therapy. And we found that RX-10 actually is highly expressed in the T cells from non-responders. This is kind of similar to a few other genes known to be negatively associated with the responsiveness to PD-1 blockade. And it's, again, RX-10 expression shows a different pattern compared to the genes associated with T cell stemness. This is another way to look at the same data sets. We clustered RX-10 with a few genes reported in the paper, either negatively or positively correlated with clinical response, and we get basically the same data. Putting everything together, I think this single cell sequencing data sets actually tell us that RX-10 expression predicts the low responsiveness to PD-1 blockade. So having said a lot, what is RX-10? What does RX-10 do in cells, especially in T cells? By definition, it is the regulator of G-protein signaling protein. Putting things in a very simple way, RX-10 binds the G-alpha protein downstream of the GPCR and accelerates the conversion from the inactive form to active form. In other words, RX-10 could be considered as a brake of the GPCR signaling. Because many chemokine receptors are chemokine receptors, therefore this molecule has, and I want to highlight that this molecule was originally cloned by Professor Lin in 1997. Because RX-10 regulates a few chemical receptors, a few immunologists have shown that both CD4 T cells and megakaryocytes migration could be regulated by RHT. But before our study, we were not able to find anything relating RHT in the CD T cells. So we thought this is pretty cool. We wanted to move on. We asked 3 questions that first, what signals induce this molecule in T cells? Is RHT only a marker, or does RHT really do something functionally in T cells? And then finally, what we can do to in favor of the clinical response. To answer the very first one, we decided to generate a mouse model to report the expression of RHT. There are several reasons. First, it's very difficult to identify a really reliable antibody for RHT. Second, the RHT is the intracellular antigen. It's not easy to stain them in live cells. So this pretty straightforward design. We put a fluorescence M-cherry after RHT. Theoretically, if a cell expresses RHT, the M-cherry would be turned on. We also put a Cray ERK2 after M-cherry. So this enables the lineage tracing. For example, if we give tamoxifen, which induces Cray activity, the Cray will cut out the 2 LuxP sites flanking the stop codon in front of the YP, and these cells will be turned on as green. So therefore, there are several possibilities. If a T cell does not express RHT, it does not show any fluorescence. But if the cell turns on RHT, it will be red. And if the cells turn on RHT expression, and if we give tamoxifen, the cells will be both red and green, so we call it yellow. If these cells in the past expresses RHT, but later lost RHT, the YP will remain, so these cells will be green. So theoretically, there are several possibilities. As a proof of concept, we treated CD cells with CD3 antibody, CD28 antibodies to induce expression of RHT. And at the beginning, we were not able to detect any RHT. But as time goes by, especially after 24 hours, we were able to identify a few cells expressing RHT. And at 65 hours after stimulation, we were able to detect a decent amount of T cells. To really give you a feel of RHT expression hour by hour, I want to show you this little movie. Hopefully, no. Yep. So anyway, it's basically the same. So I'm going to show you the movie, but it tells us that they basically the same thing, but after 1 day and 2 days, the RHT really turned on in T cells. You can imagine that this is really beautiful, a lot of M-cherry positive clusters. We also validated this M-cherry 2 mouse. We sort out the M-cherry high cells and low cells. We detect the mRNA level. We also perform mass spectrometry to validate the protein level, and it works as we would expect. We also try to identify the inducers of RHT expression, and we found that CD3 actually is a major, the most potent inducer of RHT. A few other cytokines, for example, the STAT5 activating cytokines I2 and I15 actually induce a big amount of RHT. This is very similar to the PD-1 molecule, which is included as positive control here. So we then move on to the in vivo setting, because when we threw all the antibodies in the cytokine to the cell culture, it's really artificial, it's really in vitro. What does RHT do in vivo? We want to look at the expression first in vivo. We first implanted 2 tumors in the same mice. They're both B16 melanoma, but one cell line expressing the JP33 epitope, the other one expressing OVA. And then we transfer the P14 cells, either naive or pre-activated P14 T cells. So again, those T cells could specifically recognize this epitope, but not this one. And then we recover the tumor infiltrating T cells in both tumors. color-coded here, and we found the M-cherry is pretty high in the B16 melanoma cells expressing the cognate antigen, but not the other one without its cognate antigen. We saw the same thing, similar thing in the other experimental setting. So this study tells us that the antigen stimulation does not only induce the expression of RGX16, but also maintains the RGX16 expression. I say maintains because before the adoptive transfer to the tumor-bearing mice, these T cells have been already pre-activated in vitro, and RGX16 is already turned on before the adoptive transfer. So when we transfer these cells to B16 ova, these T cells lost the expression of the M-cherry. But in the tumors expressing the cognate antigen, some of the T cells still express M-cherry, suggesting the maintenance of RGX16 also depends on the antigen stimulation. So to that we try to analyze the subsets of T cells expressing RGX16 and related that to other markers of T cell exhaustion. We found the tumor infiltrating T cells in the M-cherry mice can be roughly categorized into 4 subsets, 1, 2, 3, 4. This subset express both M-cherry and PD-1, especially if you look at the PD-1 expression itself, this population expresses highest level of PD-1, and this actually shows very similar pattern with the PD-1 versus tox staining pattern. So this is from Barry's lab, the tox paper, the famous tox paper. And we found this population actually almost undetectable in the spleen because apparently the tumor antigen is more abundant in tumor than in the spleen. This also echoes our results that are shown in the previous 2 slides. Because there are so few P4 in TILs in the analysis below, we only focused on P1, P2, and P3. Based on the color coding, I think you're able to follow this. We found the P1 cells, which express both M-cherry and PD-1, they actually express higher levels of inhibitory receptors like TIM3 and TIGIT. The same thing for CD11, CD38, and CD244, compared to the cells in P2 gates and P3 gates. To me, I think the cool thing is the expression of tox and cleaved caspase-3, which is surrogate for the cell apoptosis. We found P1 cells, they express very high levels of apoptosis marker and also tox. This kind of tells us that the RGX16 might be implicated in cell survival and apoptosis, which is also echoed by the ratio between the BCL2 and the BIM. So we are also very excited to say that the M-cherry expressing cells express lower levels of TCF1, which is this marker is known to be reciprocally expressed with a few other genes related to T cell terminal exhaustion tumor. We further categorized the expression of ELMS, TBAT, and also very importantly, KRG1 and the hormone receptor selecting CD62L, and got the general impression that the RGX16 expression positively correlates with the T cell terminal differentiation, but negatively associated with T cell stem list. So then we move one step further. We try to test whether these terminally differentiated cells still retain the capacity to generate other proteins. So this is kind of similar to Miller's paper or Heinen's paper, and actually a commonly used approach to evaluate the T cell stimulus. So we use flow cytometry to sort the tails into different subsets, and we transfer this tails into different tumor-bearing mice. So this is basically summarize of what we saw. When we transfer the R16 high cells, which is marked by high expression of M-cherry, when we transfer this M-cherry high cells, PD-1 high cells, and a few days later we recover them, the cells basically maintain the same phenotype. They didn't change. But if we transfer the PD-1 high cells, but M-cherry low cells, they actually give rise to the M-cherry high cells, suggesting that this is the precursor cell subsets, and they are able to give rise to the progeny cells. And this percentage, actually the numbers actually, of this P2 cell progeny is higher than the one from the P1, suggesting that this one has a better proliferative capacity than P1 cells. If we transfer the cells which were negative for both M-cherry and PD-1, they were able to repopulate everything. And the numbers of the donor cells actually exist, the first two groups, suggesting this group of cells retain the highest levels of T-cell stimulus. And there's a gradient of stimulus and the proliferative potential from P3, P2, and P1. And we were also able to use pie charts to show you this, this conclusion. Very excitingly, we found that the tox expression is actually pretty high in the progeny cells from P1 and P2. So this kind of tells us that this population, the M-cherry positive and PD-1 positive cells are really terminally differentiated. And second, although these cells at the beginning express lower levels of tox, but their progeny acquire this terminal differentiation phenotype, which is shown by the tox expression. So then we, as I said at the beginning, not the beginning, 2 slides earlier, we found there's a kind of implication of R16 expression in the T cell survival. Therefore, we try to look at the expression of M-cherry and whether this expression relates to T cell apoptosis in vivo. So if you still remember the introduction of the mouse model, we crossed the M-cherry mouse model with the stop codon, which is flanked by the LOXP reporter mice. So in this model, if we gave tamoxifen, which induced activity of Cui, we were able to induce Cui activity, and Cui cut out the 2 LOXP sites flanking the stop codon and release the YP expression. So as shown here, these cells, they didn't express M-cherry, and these cells, they just gain M-cherry expression, but they were they probably didn't fall into the window of the tamoxifen activity, which I will explain in the next slides. And these cells were actually, we call them early responders. So they gain expression of M-cherry very early, and they got a chance to be labeled by YLP. And then we look at the annexin 5 and also DEPI in these cells, and we found that these green cells, actually we should call them yellow cells, but it's kind of color coding in green. These cells, they express much higher levels of not expression, but the internal annexin 5 expression. And if we look at the annexin 5 2 days later on day 15, first, the population, the percentage of these cells is lower than 2 days earlier, suggesting the shrinking of this population of cells. And the annexin 5 MR5 is further decreased. This is 7,000. This is 6,000. So it's much higher than 2 days earlier. We also look at 2 days later on day 17. On that day, the percentage is further shrinking, and these cells became DEPI positive, suggesting death. So according to the literature, if we apply tamoxifen on day 0, 2, and 4, on day 11, which is 1 week after the last injection of tamoxifen, the concentrations of tamoxifen drops back to the normal level or drop below the detection limit. So therefore, this is the estimated window of reactivity. So only if a cell expressing M-cherry during this window will be labeled with M-cherry and VIP. When we take down the mice on day 17 of the tumor cell implantation, which is actually 6 days after the end of tamoxifen window, these cells die. So based on this very simple mathematical model, we estimated that as early as 6 days after acquisition of the M-cherry expression, these cells undergo apoptosis in the cell death. But because the earliest possible time point to acquire the expression of M-cherry and VIP is day 0. So the death time wouldn't exceed 17 days. So this is very, I would say, oversimplified model to estimate how long the exhaust T cells could survive in a tumor microenvironment. It might differ from one model to the other one. Having seen this overlapping between RD16 expression and markers with T cell exhaustion, we wanted to move one step further to look at whether RD16 really regulates T cell exhaustion. So we used the RD16 deficient mice. We categorized their immune cells before implanting the tumor first. In general, this deficient mice develop very comparable thermocytes compared to the wild-type peers, suggesting RD16 does not regulate the thermocyte development. The only phenotype that we saw in the steady status is there are more RD16 deficient lymphocytes and splenocytes, suggesting that RD16 might suppress naive cell homeostasis. And I wanted to highlight that this difference between RD16 deficient mice and PD-1 deficient mice. PD-1 deficient mice will develop autoimmune disease when they get older, but this seems not the case for RD16 deficient mice. We let the mice age almost 1.5 years, but they're totally fine. We were not able to detect any infiltrates in any organ. And furthermore, the cell surface markers like alpha-lectin, CD44, CD69, and IL-7 receptor, they all look comparable between vitamin and KL, suggesting that the RD16 and KO mice are doing really good at the steady status. So before we implant tumors, we also visited the literature, and we found that in general, this TCR-induced genes like PD-1 tox deficiency could trigger different outcomes. The PD-1 deficient mice, for example, develop much smaller tumors. But if you completely kick out tox, these cells will die. So they still need one copy to maintain their survival. So one copy, the cells with one copy of tox, they're able to kill tumors much better. So we were very excited whether RD16 follows this trend or this trend. We got an answer pretty soon. So we kick out RD16 and we found this mice, we implanted B16 tumor cells to the wild-type and KL littermates, and we found the KL mice develop much smaller tumors compared to their wild-type peers. We used different ways of following the reviewers' comments. And we were also able to find the leukocytes numbers and the density, and also CD8 T cells density in the tumor. They're all increased by the RD16 deficiency. Due to the time limit, I didn't show you other data, but I can tell you the punchline is the RD16 deficiency also increases the CD8 T cell survival in tumor. This is also accompanied by increased effector cytokine production, suggesting that RD16 actually shows kind of different phenotype with the tox KO mice, although they share similar expression patterns. So then we try to ask why, what is the underlying mechanism? We screen a few signals downstream T cell receptor, both proximal signal and distal signals. And eventually we narrowed down to ERK phosphorylation. We found the auristatin deficiency increases the phosphorylation of ERK. So I have to skip quite a few experiments because this really take several years to narrow down to ERK. We then ask how auristatin regulates ERK phosphorylation. We used a very old-fashioned approach. Briefly, we activate CD T cell in vitro. We then overexpress auristatin, which is tagged by flag. We then pull down the auristatin protein, and then we resolve this protein using SDS page, and we submit this immunoprecipitates to EMBL to identify the potential interacting proteins. We found one of the hits actually was identified in this gel block. It's called RQGAP1. It's kind of platform molecule. According to the literature, RQGAP1 provided a platform for RAF and MEG ERK signaling cascade. And this platform molecule facilitates this phosphorylation of the effector molecule. So then we ask whether auristatin interacting with RQGAP1 also influences the downstream of ERK phosphorylation. First, we were able to validate the mass spec data. When we pull down auristatin, we were able to detect RQGAP1 in the Western blot. We also compared the expression and pull down efficiency of the RAS-RAF signaling cascade. We found that the interaction between RAS and RQGAP1 in the white cells pretty weak. This actually was also reported in the literature. But the auristatin deficiency strengthened the interaction between RAS and RQGAP1. We saw a similar thing for RAF. But the expression of RQGAP1 between white and killer cells, they're actually not that much different. So this data tells us that the auristatin probably enhances ERK phosphorylation by strengthening the interaction between the upstream activator proteins with this platform molecule. Yeah, this is pretty much what I just said. I think I still have 15 minutes. I will try to wrap up before the end. We then ask whether there's a causal relationship between the enhanced ERK phosphorylation and the decreased tumor growth. To do this, we designed a kind of complicated experiment. First, we implanted the B6 mice with this B16 tumor cells expressing this tag, this epitope. And then we transfer either wild type or R16 deficient P protein cells to these mice. And we engineered these cells by overexpressing either wild type ERK or the ERK that couldn't trigger downstream molecule. You can consider this as dominant negative. So there are two ERK forms, ERK1 and ERK2. We didn't choose ERK2 because according to previous study, ERK2 deficiency influenced T cell function. So even we saw the rescue using ERK2, we couldn't attribute this to the RGX10 intrinsic role. So that's why we choose ERK1. So first, we found the deficiency of auristatin still decreases the tumor growth. That's great. But when the ERK is, when we transduce the T cells with the dominant negative ERK, auristatin deficiency no longer increased the tumor control or slow down tumor growth. So this might be difficult to follow, but in general, it tells us that auristatin deficiency slow down tumor growth by enhancing the ERK, especially ERK1 activity. We also monitored the tumor weight and the donor cell numbers, density, and their phenotypes. Due to time limit, I wouldn't go into that. This is kind of cool, but the reviewers want more. They ask whether auristatin deficiency shows any synergy with the PD-1 blockade. And this is actually what we wanted to do. First, we implanted the RT wild type in the KO mice with B16 melanoma cells. Again, the deficiency of RT decreases tumor growth. When we treated the wild type mice with PD-L1 blocking antibody, we didn't say decrease tumor growth. This echoes the reference showing the B16 tumor cells, they're kind of resistant to the PD-1, PD-L1 blockade. But R16 deficiency sensitizes this melanoma to PD-L1 deficiency. This kind of, at least the combination of effects between the two, or you can even say there's synergy between R16 deficiency and PD-L1 blockade. This is the way that we think how it may work. First, in the presence of the chronic antigen, the T cell receptor turns on fill signals, both proximal and distal signals. Both R16 and the tox, I think also including other proteins, other genes, they're kind of depending on the mFAT, mTOR signaling. And in terms of R16, it inhibits ERK and deficiency of RDX16 enhances the cell survival and the effector function. We also learned a few things from this story. First, RDX16 is not only a marker to predict the responsiveness to PD-1, PD-L1 blockade, but it also functionally inhibits T cell responses and their sensitivity to PD-1 blockade. I think this study also opens several new threads. According to the tox papers, the expression of tox is shows pretty different pattern between the tumor microenvironment and the chronic infection models. For example, in the Schillinger's paper, she showed that the tox high cells express very low levels of T cell 1. In T cell 1 high cells, they're actually low in tox. But according to Professor Dimmerzen's paper, tox high cells actually express high levels of T cell 1. So apparently, the correlation between T cell 1 and tox is probably model specific. We also want to ask whether RDX16 does something in the infection model. To do this, we first use the acute infection model on strong, which I introduced at the very beginning. And very interestingly, we didn't find anything. So RDX16 deficient mice, they're doing pretty similar compared to their wild-type peers. Their subsets which tell their fates of either effector cells or memory cell, they're also comparable between the two. The only trivial difference between the wild and the KO is there's slightly increased cytokine production in the KO. So far, we are planning the chronic viral infection model and trying to see whether RDX16 regulates T cell exhaustion also in the chronic infection. So I want to thank our team. This is this fantastic team. And this work is mainly done by very talented PhD student, Nina Voshan, and she already left the lab and now works in the Guoliang, the BI company, the pharmaceutical company. And I also want to thank the general financial support from the foundations, in particular the Deutsche Krebshilfe Foundation and also the Hecht Foundation and the Cancer Research Center in New York. And very recently, the European Research Council. So this is pretty much what I want to say. And I want to stop here, and I'm ready for questions. Thank you very much.